The ways in which a hormone affects a cell are dictated by their chemistry. In general, hormones fall into one of two categories, based on whether they're soluble in lipids or soluble in water. In other words, lipophilic or hydrophilic. Hormones synthesized from lipids, like steroid hormones, are lipophilic. They can cross the plasma membranes of the cell and nucleus, but since they're not water-soluble, they need to bind to special transport molecules in order to travel through the aqueous medium of the bloodstream before they reach their target cells. Once there, they can cross into the nucleus, where they most often serve as transcription factors that control gene expression and protein synthesis. Hormones synthesized from proteins, with the exception of the thyroid hormones, are hydrophilic, and therefore have no problem traveling through the bloodstream when they're secreted by endocrine cells. However, they can't cross the plasma membrane when they reach their target cell, since they aren't lipid soluble. So to achieve their intracellular effects, cells use multiple classes of transmembrane receptors, the most important of which are the G-protein coupled receptors. These proteins have a lipophilic middle segment that allows them to remain embedded in the plasma membrane, with one hydrophilic end protruding into the cytoplasm and one hydrophilic end sticking out of the cell. Now that extracellular portion is shaped almost perfectly to bind to a specific hydrophilic hormone, but not exactly perfectly. The binding site on the receptor fits the hormone well enough to where, when it gets close, the hormone becomes attracted to the receptor, like a tiny magnet. But the receptor is attracted to the hormone as well, and flexible portions of the receptor warp and twist to grab onto the hormone more tightly in a process known as conformation change. And because the receptor stretches across the entire plasma membrane, the conformation change induced by binding on the extracellular side will alter the shape of the cytoplasmic portion of the receptor as well. What happens next varies based on the class of receptor, but this is the basic principle of how an extracellular signaling molecule causes changes to the inside of a cell. In the example of the G-protein coupled receptor, or GPCR, the receptor is bound to an inactive G-protein trimer and blocks a binding site on the G-protein that allows the trimer to dissociate and free itself from the receptor. The conformation change exposes that site, allowing the G-protein to detach into a cytoplasmic and a membrane-bound portion. Each activated G-protein unit then goes on to activate many more enzymes within the cell, which can then activate even more proteins and channels, so that the signal that began with a single binding event is amplified until hundreds of intracellular molecules and channels are then activated. And because of this adaptation to their chemistry and their inability to cross a plasma membrane, hydrophilic hormones are most frequently the ones that cause very rapid, dramatic changes in cells that can nonetheless be quite short-lived. Think hormones like epinephrine, for example. Conversely, lipophilic hormones, which are poorly water-soluble but can move through membranes directly into the nucleus, tend to exert their effects by directing the de novo transcription of new proteins, rather than activating existing enzymes. These are often slower to act, like the sex steroids, but given time, can exert major structural effects on cells and tissues that tend to be more long-lived.